Okay, so I'm going to be giving two presentations in one. So the first part, I'll be focusing on sex differences in HIV cure research, and in the second part, I will be, I'll be talking about the status of women's participation in current cure-related studies. And I would like to begin with my acknowledgements by thanking Eileen Scully, Sarah Gianella, Katie Godfrey, Richard Jeffries, Danielle Campbell, and Karine Dubé, who have shared many slides and much information with me to develop this talk, as well as Treatment Action Group, the ACTG Community, Women's HIV Research Collaborative, and ATAC. So there is one slide in here from a larger slide deck that the Women's HIV Research Collaborative produced, which is a great resource if people need to seek out more information. So thank you all for also Karine, Richard, Danielle for figuring out the technology to keep this going, and Michael, of course. So to start with sex differences in HIV cure research, and also to give a teaser, spoiler for, not a spoiler, but a teaser. So Dr. Eileen Scully is giving a talk on Tuesday morning of CROI about this very topic. So watch the video cast of that when it is released, or if you are registered for CROI, you can watch it live, I would imagine. So she'll give probably much more of a scientific overview than I will. I suspect many of us are aware that the global burden of HIV infection is female. So 18.8 million women and girls, which is somewhere around 50% of people living with HIV worldwide. We are making progress with those numbers, so death rates are declining. It's sort of globally, AIDS-related illnesses are the leading cause of death among women and girls ages 15 to 49. Young women are 2 times as likely as young men to acquire HIV. This is globally. It, of course, will look different in different countries. And 3 of 4 new HIV infections are in 15 to 19-year-olds. I'm sorry, 3 of 4 new infections in 15 to 19-year-olds in sub-Saharan Africa are in girls. So the global burden of HIV infection, as you can see, is really shifting. Associated with HIV are issues like intimate partner violence, which increases women and girls' risk for acquiring HIV and also disproportionately affects women and girls. And so it's not just HIV, but also overlapping epidemics. In the United States and in Baltimore, where I live and where Eileen Scully lives, women account for 20% of cumulative AIDS diagnoses and are 23% of people living with HIV. And so this map here, you can see the darker the red, the higher the prevalence of HIV with all sexes and genders, and then just in women. And to speak about transgender women and HIV in the United States, the HIV prevalence among trans women in the US is much higher than in the general population. So general population HIV prevalence is 0.5%, and in transgender women, it's 18.8%. If you break this 18.8% down by race and ethnicity, US transgender women are predominantly Black or African American, as well as Hispanic, Latinx women, or I'm sorry, white and other. And so, again, social structural issues are driving HIV rates disproportionately to reflect other sort of social marginalizations. But then to think about biological sex and why that is going to be relevant to HIV cure. And so the literature tends to use biological sex and sex instead of sex assigned at birth. And so you'll see that reflected here. And I'll talk a bit about some of the characteristics that are included in this category of sex assigned at birth or biological sex. So as I've just told you, and this is an analysis from Rowena Johnston, the burden of infection, pink for women, blue for men, is about 50-50. enrollment in all cure trials, you can see, is much more skewed towards male participants than female participants. And as I'll talk about a bit later, even this skewed ratio will vary depending on what type of cure trial. So in, for example, latency reversing agent trials, it's even more skewed. And so, thinking about some of the biological determinants of sex differences, there's going to be anatomic compartments, which I'll talk about in a bit, genetic variation, immune cell phenotypes, epigenetics, and the microbiome. So hormonal differences will also be important to consider. So both hormones that are made by the body and also hormones that are introduced into the body, what are called exogenous, so whether that's done for any number of different therapeutic reasons or gender affirmation therapy. For genetic differences, it's important to consider the X chromosome carries significantly more genetic material, and it's also bigger than the Y chromosome. And so there are estimates that the X chromosome carries between 6 and 10% of all microRNAs, along with immune regulatory genes like toll-like receptors. As far as immune responses, females have a more robust immune response than males, and tend to respond better to vaccines. And you can see some interesting data. For example, right now, people are talking about coronavirus, and females are having better outcomes than males with that, just reflective of that more robust immune response. There's also going to be differences in the epigenetic landscape, differences in establishing latency. So women who are assigned female at birth tend to have smaller HIV reservoir sizes than males. And then I'm going to talk on the next in a few slides about microbiome factors. So but the point is that there's, biological sex is not just XY chromosome. So there's a number of different variances. Each of these matter when designing curative strategies as the endpoint and also along the way of getting there. So in HIV cure research, we see measured differences in viral persistence, in relationship to T cell parameters, and in transcriptional controls. We also need to keep in mind that the therapeutic targets of HIV cure research are affecting the person, right? And so thinking about all of those ways that I showed you on the previous slide that are components of biological sex, all of those are implicated in curative strategies. And so thinking about differences in immune pathogenesis, so there's a complex relationship between sex hormones and the immune system. You may be aware that, so for example, during pregnancy, the pregnant person has a compromised immune system, partly because progestins will suppress the immune system in order to preserve the pregnancy. There's fluctuations within the menstrual cycle, fluctuations across the life course at what's called reproductive transition, so at puberty, at menopause, during pregnancy, post-pregnancy. Our understanding of the relationship between exogenous hormones and the immune system is also limited. So most of the information we have about hormone therapies and their impact on the immune system comes in the case in like postmenopausal women on hormone therapy. So we have very limited data on people taking hormones for gender affirming therapy and the impacts of that on the immune system. You'll have to excuse me if I'm jumping between two computers so I can see my notes. So I keep clicking the wrong one. So, we need to just keep in mind that the sex hormones will play a role in cure strategies. There are differences between the sexes in HIV reservoirs, both in tissues and in anatomic compartments. So we know that HIV is not distributed evenly. It's higher in the gut compared to the blood, but it will vary even across the gut. So one location in a person's gut might have different distribution, higher or lower distribution of HIV DNA than another location. There's also differences between the sexes on all of these components that I've been talking about. So hormonal, metabolic, fat distribution, immune responses, and PK differences, so how drugs are metabolized. And then we know that sex hormones play a role in HIV transcription. And so these factors combined could mean that the location and amount of replication competent HIV will be different in men and women, and that these differences may be relevant to future curative strategies. Thinking about the female genital tract, which I have abbreviated as FGT on the first and fourth bullet point, that, like many aspects of the female body, is complex. There are different compartments of the female genital tract, and there is no equivalent of these compartments for males. So both the upper female genital tract, which includes the uterus and the fallopian tubes, as well as the lower female genital tract, which would be the vagina and the lower cervix, can have features that, or do have features that allow for pregnancy, but also change the risk environment for HIV acquisition. And so it could be important to consider those different compartments. Also, that there's a difference between the upper and lower compartment, as well as the non-existence of those compartments in males. So when we're trying to characterize the HIV reservoir, which is a component of HIV cure research. There is much that remains unknown about the amounts of replication competent HIV, the relationship between blood and the female reproductive tract. There's evidence that even if somebody's HIV RNA levels are undetectable in blood plasma, there's ongoing virus production in the genital tract. And so the takeaway is that each part of the female genital tract has a role in maintaining reservoirs and the immune activation and HIV progression, but this is an area of research that we just don't know a ton about yet, or enough about. Thinking about specifically the vaginal and the neovaginal microbiome. So we do know that the vaginal microbiome plays a role in risk of HIV acquisition and transmission, as well as STIs, other infections and diseases. We also know that there's variation in the composition of an individual's microbiome across the life course and during reproductive transitions. There's also variation across different communities and geographically in the composition of the microbiome, because this is impacted by a number of external factors, including the things you eat, the chemicals that you're exposed to, etc. There is limited knowledge about the neovaginal microbiome. We do know that the composition of that microbiome will change depending on the source. So a natal vaginal microbiome tends to be, when healthy, is dominated by lactobacilli kinds of bacteria. And that's what's most commonly found in reproductive age women. In a neovaginal microbiome, there are those same kinds of bacteria if the source of the neovagina had been penile, but if it is sigmoid colon source, then there could be different kinds of bacteria. And so this is just an area that we know insufficient information about. I can see that. I think maybe there are questions coming in, but I'm going to pause after this part and answer all the questions is my plan, because otherwise I'll confuse myself. So some additional considerations for cure research with women. Collecting sex-specific samples can be a challenge. So adding a pelvic exam into research can add time and be inconvenient both for the participant and for the provider. If you're going to account for all of the variations that can occur just within the menstrual cycle, so just within that approximately 28-day period, you would need to take samples throughout that cycle in order to draw really robust conclusions. Self-collection is an option, but not all women are comfortable with that. So some studies I know within the ACTG are probably elsewhere are moving towards when a vaginal swab is needed, asking the participant or allowing the participant to collect the sample themselves, but not everybody is comfortable doing that. And keeping in mind that there's never enough money, and studies always want to enroll faster, which can lead sites to rely on established recruitment practices or draw from the same pools of potential participants. A number of barriers to engaging women. So within the ACTG, the AIDS Clinical Trials Group, the precursors to the current Women's Enrollment Initiative, which is now called Team Women and being spearheaded by Danielle Kimball, thank you. In 2014, that group did a survey of ACTG community members about why women are not participating in ACTG trials and cure research. 33% of community members said competing priorities, 13% indicated cultural stigma and a lack of appropriate incentives. 14% felt that a lack of education about the clinical trials process and lack of awareness about current trials was a barrier. 8% unwilling to serve as a guinea pig, and 8% felt that recruitment strategies lack geographic and demographic diversity. So, I think most of us on this call, if not all of us, are really aware of these issues and more, right? I expect these numbers would look slightly different if we resurveyed in 2020, but we do know the issues, and it's just a matter of addressing them. The implications of not engaging women in cure research, of differently engaging women in cure research. If we are able to do not just overall analyses of a strategy, but sex disaggregated analyses, we could have novel insights. Some of these immune therapies should be considered, especially in light of known sex differences in immune pathways. And whenever we're measuring viral rebound or the reservoir size, we need to keep in mind that there are sex differences that likely do matter. If I'm able to speak with certainty, I would say that they definitely do matter. And, keeping in mind as well that the global burden of HIV in women obligates that cure research speaks to these sex differences, right? And so it's not just more interesting science, right? But it's also who is the cure research going to be for? And if you think back to the maps that I showed, or the numbers that I showed at the start of this presentation, women are going to need curative strategies, and not just women in the US. And so we need to really be pushing for sex and gender and geographic diversity, which takes us to the second part of my talk. Danielle or Michael, do you want me to take questions now or keep going?
Keep going, Liz. We'll take them at the end of your talk.
Okay.
As we progress.
Okay. So I was able to work with Richard Jeffries and Treatment Action Group to do a second landscape analysis of current HIV cure research in 2019. So we had first done this landscape analysis in 2018 and presented those results at this workshop in Seattle last year. And those results also were published in Journal of Virus Eradication, and there's a community-oriented write-up available on TAG's webpage. So we were able to repeat the survey and the analysis again at the end of last year, so like the fall of last year. And just to give you just a brief description of the methods, and then I'll kind of turn to the demographics and the overall landscape. So at the time of our survey last year, there were 133 studies on the TAG listing, so the Research Towards a Cure section of the TAG website that Richard keeps meticulously updated. So when we started the survey, there were 133 studies, and we received some kind of response from 73 studies. So we contacted PIs, study coordinators, site contact, the person listed in clinicaltrials.gov as the contact, somebody we might have known personally. We contacted someone for all of these studies and received 73 responses. So 5 studies declined to complete our survey for a number of reasons that you can see here. One study closed during our survey period, like stopped development, not close to accrual. 2 people responded that their study isn't actually really a clinical trial, isn't really a study, and so it wasn't appropriate to answer the questions. And then 65 people did complete a very lengthy survey that we sent to them, for which we are truly appreciative. 37 of those 65 had also completed our 2018 survey. So it's about a 55% response rate, if I round up, which is similar to the response rate that we had in 2018. And these results will be hopefully interesting to everyone. So as I said, 133 studies on the tag listing in August of 2019 in 24 different categories. So thinking about the HIV cure-related research landscape, there are a variety of curative strategies that are being studied. So it's not just, we've heard about a number of them today, and we'll continue to hear about more of them. But likely each one of these categories has some sex differences may be relevant in any or all of these categories. And so you can see here, what Richard and I did is we got some information from the data available on different registries, so clinicaltrials.gov, but also other trial registries. And we were able to determine the projected number of participants by category of trial. So when there's more than one trial, we've given the mean and the range as well. I recognize this is a lot of numbers. I believe the slides are going to be made available. I don't want to read you all the numbers, but I want to point out that the mean number of participants is pretty low here. It's unlikely that you'll be able to make a claim about sex differences in a study that has an N of 5. However, this is where we can think about things like sort of doing a meta-analysis later, or data sharing as perhaps a way to answer questions. So if we were to identify a set of questions that are sex-specific or relevant to sex, that could be answered then by pooling data or looking across studies. I also want to note that there are enrollment trials that enrollment targets for this area. So one of these studies, and then also one of the combination studies, represent studies that are going to be, so the actual study is looking at HIV treatment in newborns, but they'll be enrolling the pregnant person, and so that's why the N is so high, because they expect that only 5 to 10% of the newborns will actually be enrolled into the study, so they're not actually enrolling 905 people to continue in the study. And then this information here, so current enrollment by category, this comes from the survey responses that we received. So we asked the people who took the survey if they were able to estimate for us, or give us actually a specific number of how many people had been enrolled to date in their study. And so you can see that it's still low. So the numbers of people enrolled are still very much in early stages, not just of as far as phase of research, but also of the actual studies. So the range of enrollment 3, 1, 3, going as high as, again, for this observational study, 624. Some information that I think is interesting to look at. So here you can see we asked survey responders what countries their trial was enrolling in. So you can see that most studies are enrolling in the United States, a number of studies in Thailand, a number in Europe, China, South America, and African countries as well. But of our responders in 2019, the overwhelming majority are enrolling in the United States. The blue line on this graph shows where all of the studies on the tag listing are enrolling. So the purpose of this illustration is to show that our respondents were relatively representative of all studies, right? So the tag listing is all cure-related studies happening, and then our respondents were representative proportionally to where the trials are taking place. So yes, most of the respondents were conducting studies that are enrolling in the United States, but also most of the studies are enrolling in the United States. So that's an important thing to consider going forward. And that the geographic distribution, so again, this is all studies from 2018 on the yellow line to 2019 in the blue line. Studies, the geographic distribution of studies has not shifted. So we're still seeing most studies taking place in European countries, in Thailand, and in the US, which might be an area of advocacy going forward, in my opinion. And just to continue characterizing the studies that were taking place last year, because I suspect this group would be interested in that. Most studies are enrolling participants who are on antiretroviral therapy and virally suppressed. So that's this blue bar here. Oh. One study is enrolling participants who are treatment naive and will be required to remain off ART. A number are initiating treatment during the study, and then also some have different criteria. So you can see most of the studies are enrolling people who are virally suppressed. This figure here shows the invasive procedures that are happening in the different studies. The lighter peach orange color is when a procedure is optional, and the darker color is when a procedure is required. So lots of leukapheresis, lots of ATIs, lots of chemo. And, I mean, I guess lots is not the most scientific term, but you can see the taller bars for leukapheresis, ATIs, biopsies, lumbar punctures. And so, you know, that's fantastic potential sources of data that could be used to identify, explore, and characterize sex differences should sufficient numbers of women be enrolled. And so then I want to turn to the demographics. And I want to say just a note about some relevant US regulations. So we hear often about the NIH Revitalization Act of 1993, which is when the NIH, well, when Congress made it law that the NIH-funded research had to include women and minority groups in all NIH-funded clinical research in a way that is both appropriate to the question under study and will allow a valid analysis of whether the variables being studied affect women and minorities differently. And this has been updated a few times, like in 2001. And you know, results are supposed to be disaggregated and reported by sex, as well as racial and ethnic category. And this regulation is applicable to phase 3 studies and pivotal phase 2 and 4 studies, right? And as I'll show you in a minute, the majority of HIV cure research is not phase 3 research. So this does not apply. Oh my goodness, that should say 2014. But the NIH has also implemented a policy on sex as a biological variable. So this, unlike the Revitalization Act, is not a law, but it is an NIH policy that NIH expects sex as a biological variable will be factored into research designs, analyses, and reporting in vertebrate, animal, and human studies. So this is all research funded by NIH, not just phase 3 trials. This is really aimed at basic and preclinical research. In this new policy, there needs to be a strong justification provided for applications that are proposing to study only one sex. And again, this is for NIH-funded research. Oh, there's my animation. Okay, that's happening. So this figure shows of the 2019 responders to the survey that Richard and I did, who did tell us their funding source for studies. The green studies received at least more than 0%, so at least some funding from the US government, which is 60%, but 40% of studies did not receive any funding from the US government. So what that means is that the NIH Revitalization Act doesn't apply to them. The SABV policy doesn't apply to them because they're not receiving US government funding. And so that's important to keep in mind. Another thing to keep in mind is that cure-related research is largely early phase. So this graph comes from the clinicaltrials.gov and other registries, showing all of the studies by phase. And you can see these are the studies that definitely do and may fall under the NIH Revitalization Act. So the studies over here would fall under the SABV policy, but this is not, this is mostly phase 1 research, and so not applicable to the same regulations. And even the sex as a biological variable policy that was established in 2014, I think took effect 2015 or 16, and so that is still sort of gaining traction in the collective consciousness of the research world. And so moving on to the results as far as demographics, the survey that we sent asked, does the study have any of the following demographic targets beyond what is required by inclusion and exclusion criteria? And we asked people to specify if they had a target on these categories, so percentage female, percentage women, to get at both sex and gender, percentage over 50, percentage of Hispanic descent, Black or African descent, Asian descent, and any other. Danielle, am I okay on time? I think I'm not, but
You're
I can
okay.
try. Okay, I can talk quicker if I need to.
No, this is great.
This is out of.
Can you hear me, Liz?
Yes.
One of the questions that was asked is, how did you target your survey results, and were study participants included?
Sure. So this survey went to the representatives of the trial. So a PI, a study coordinator, a co-investigator. So this survey went to the researchers, not the community members or participants. Most of the respondents were PIs or investigators, but a couple were study coordinators responding on behalf of the study team. So, and because we're asking about studies that are currently in development and enrolling, so one, we can't really we would have no way of getting in touch with participants in a current study other than through personal connections and happening to know where someone might be. But we wanted to do this kind of real-time categorization of what the landscape is right now. Does that answer the question? Okay, so the demographic targets, again, you can see the percentage of, so not everyone answered every question in the survey, which is why we've given the percentages. What is important, this is probably the most important row here, that 76% of people who answered this question said their study did not have a target, formal or informal. For sex, 86% had no formal or informal target for gender, formal or informal targets for age, or for race or ethnicity, right? So most studies have neither formal nor informal targets, is the takeaway on this slide. 60 respondents provided information about current enrollment. And again, not every question was required. So some people only told us about sex, some people only told us about race or ethnicity. But this slide will break down some of those responses a bit. So of the total participants who are currently enrolled, this is a snapshot last October-ish of current enrollment in HIV cure-related research. So 16.7% of participants are female, 18.5% of participants are women, 1.4% of participants identified themselves as transgender to whoever was collecting the data for the study, and 6.7% were over 50. So this does not match the demographics of HIV globally or in the US. And we also, as I said, asked people to provide information about current enrollment of race and ethnicity. And you can see 617 of the 629 Asian participants are enrolled in studies in Thailand. And so if you take those out, then the, excuse me, I'm clicking too fast, then this is a more accurate breakdown here. So 5.5% Asian. And to get to the question about what to do next and who we surveyed for this. So the results of this write-up, this full write-up was submitted to the Journal of Virus Eradication, which published the first landscape analysis, and we have a community write-up that will be shared after the article is published. And we hope to do some follow-up engaging with both the study representatives and community about community outreach plans, so how plans are determined and monitored, about studies plans or openness to data sharing, about the reasons for not setting formal or informal demographic targets, about partner prep provision and ATI trials, and how decisions on compensation are made. So one way to continue doing this might be through focus groups or panel discussions with researchers and community advocates. We would love to do follow-up interviews both with the survey respondents about some of those questions. A number of respondents indicated they were open to a follow-up interview. And then also doing interviews with community advocates to develop recommendations and guidance for both funders, but also for advocates. You know, because it's not just the fault of the investigators or the trial sponsors, right? Like if we're asking, and this is just me talking, so I'm not representing Richard's opinion here either. I hope, well, I don't know Richard's opinion on this, but you know, if we're asking investigators and trial sponsors to reframe their questions from sort of like, why don't women participate, to how can we be more inclusive, right? So it's kind of shifting the blame from women not joining studies to studies not welcoming women. I think we really need to ask the same questions of ourselves. So do the highly mobilized communities of advocates reflect the communities that are most impacted by HIV? And if not, how can we as advocates do better to create space that centers women's voices? And that means women of color, that means transgender and gender nonconforming folks, that means transgender women of color, and you know, and so really thinking about how as advocates we are doing the outreach and the education to diversify our own ranks so that our voices that do come to the table can be more diverse, you know. And so I know there's time later today to think about intercab networking, you know, but it's really critical for the outward facing work that cabs do to like engage, and if not engage, be an ally for all people who are going to need to benefit from curative strategies. I don't know, so it's easy to be like, oh, the study didn't enroll any women, but then if I look around a community meeting and there's like no women there either, it doesn't make me happy. And I think that I'm probably over time and now just going on a rant. So thank you. And I can stop sharing and take questions if there's time.
Awesome. For such a great presentation, Liz. Anecdotes and all. Time be damned, we do have a couple questions from folks in the audience. Let's start with a very good question from Michael Luella, who asked, how do we advocate to have studies enroll African nations? What can community do? And that was, I guess, sort of echoed and seconded by B, by way of Jessica Salzwedo. She says, what can we do as African countries to help enroll such studies, since it seems as though the world is behind on researching women?
Yeah, those are great questions. And, oh, and Richard's making comments too. I don't know. Okay. So, you know, thinking about, part of it is logistic, right? So the presentation earlier today talking about a study that's a $500,000 therapy. So if there's the infrastructure and if the infrastructure is not in place to conduct a study, then the study cannot be conducted, right, without the infrastructure. And so, but that doesn't mean that the community voices can't be present still through the development, right? So it's really, I think, the inter-cab communication is something that's important during study development. So for example, in the ACTG, right, there's a community member involved in the development of every study of the ACTG on the team from the beginning. And so those community members, when they are aware of issues, can bring them to the table, right? And so, like, it was really cool when I was on the ACTG community group, and I saw a lot of guys get on board the issue of women's enrollment. And I was like, I guess not surprised. I was pleased to see that, because once they kind of cared about the issue, they were totally willing to go to bat and advocate for it. And so in the same vein, US-based advocates and European-based advocates can go, use their voice or their position at a table to be an advocate for someone whose position is not at the table. And then at the same time, really be pushing funders and investigators to ask questions that are going to require geographic diversity or sex diversity or racial or ethnic diversity to answer them. So if you don't have a primary or secondary objective that, like, if you don't ask the question, you can't get the answer. And so it's a matter of asking the questions that community identifies as being important, and people will find the money, they will find the resources, they will find the outreach materials if they have to do it. And so, I don't think it's going to be kind of a one-pronged approach, but I try to think about what are the things that I can do to educate myself about issues that I do not personally experience, so that I can be an ally and an advocate in that situation. And then how can I get people who do not experience my issues to be my advocate and ally?
Great point. And additionally, from Rico Boone, fantastic point regarding the diversity of advocacy and trials. I think that part of this discussion is that, participants in trials are not also part of the advocate pool. So definitely want to echo Rico's big ups to you about speaking to what it means to be someone affected or living with HIV and out in this advocacy space. And when we enter, I guess, some of those upper echelon meetings that happen before the meetings, and there's no representation of folk who look like us around the table, even though we are widely impacted by this epidemic, I think those sentiments should be transient in the conversations that we have moving forward. So thank you very much for mentioning that.
Yeah. I mean, I think, like, I don't know, as advocates, every time that we make a victory, whether it's getting, invited to a meeting or getting, something we want on the scientific agenda, like, every time there's progress, we always need to be looking around and saying, like, where can we do better? Where can we do more? And I believe if I'm not uncomfortable, I'm not growing, I don't know. But yeah, I mean, I think it's really important to not say, oh, we're going to get this first, and then, like, we're going to get the cure and then expand it to include women, and then expand it to include people in African countries, right? Like, no, it needs to be there from the beginning. And a unified voice from advocates demanding that is really important. Again, just my unsolicited opinion.
Definitely well received in this space, honey. Well received. Just scrolling really quickly through the chat box to be sure that we've addressed all folks' questions. Michael did have a question about appropriate incentives for women to participate in clinical trials. I think you can touch on that very, very briefly in the interest of time.
Sure.
But also keeping in mind that the beat cab will also touch that subject as well. So really quickly, 2 minutes tops.
Yeah.
What do you think?
So that's an area that we would like to do more kind of follow-up analysis on from the survey results. But I do think it was interesting that most people who responded, again, these are study representatives, not community members, said they believed the compensation or incentives provided to participants was similar to other studies in the area. And they also mostly said that the study team or the funder or the IRB determined compensation. There was not many people who selected the answer that it was determined with input from community. And so that is absolutely a conversation that needs to continue. But also, Karine has done great work about thinking about what do we mean by benefit, right, which she'll talk about, but also what do we mean by compensation. And so we think about compensation as also being engaged in care, receiving referrals for, these sort of external to the study itself. So thinking more broadly about compensation as more than just like a $20 Visa card. Thank you.